Hello everyone, I'm Divyan and I'm going to be explaining to you guys how I built a special cell that can kill COVID-19. So this special cell is called a CAR-NK cell. CAR stands for chimeric antigen receptor and NK stands for natural killer. So this chimeric antigen receptor natural killer cell is able to kill COVID-19 because it has a specialized receptor that has been genetically engineered to fight off COVID-19. So I built this receptor using Benchling, which is a gene database tool that allows scientists to effectively organize and display their genetic information. So I use Benchling to design this genetic structure for this receptor. Now, usually Benchling costs a lot of money to have, but actually they provide this resource for free for students. So I was able to get this for free. Thank you, Benchling. And so I'm just gonna walk you through the steps on how I built this and what the process was. So first, we're gonna start with the first part of our sequence, which is the CMV promoter. So the promoter basically tells the NK cell that it needs to produce this receptor a lot. So by increasing the amount of receptors on the NK cell, it just increases the chance that it's going to detect an infection. So after this, you have something special called the CD8A signaling peptide. So the signaling peptide is basically instructions for the receptor on how to be packaged and where it should go. And this basically is really important because you don't want this receptor just lying inside the cell, right? You want it to be outside the cell detecting other things, right? So basically, this comes from another protein called CD8A. So CD8A is another type of receptor that is really helpful. So we use that because it's going to be related to the CAR because they're both receptors, right? So they'll both end up in the same place outside the cell. So that's why we use this. And then the next part is the SCFV, and this part I've highlighted is just really long, is the heavy chain. So the SCFV is really the most important part of the CAR because that's the part that's going to be detecting the COVID-19 proteins. So these COVID-19 proteins called spike proteins will be detected by the SCFV, which is going to tell the NK cell to release certain chemicals to kill that COVID-19 infected cell. So this heavy chain is the first part of the SCFV, and it's going to be continued by the Whitlow 218 linker. So this linker will attach the heavy chain to the light chain, which I'm going to show you guys in a sec, and it really provides more flexibility to the SCFV, and it allows it to actually function like an actual antibody, which is what it's based on. And then it's called the Whitlow 218 because it's named after the scientist that found this. And the next part of our SCFV is the light chain, which I've talked about here. So this is the sequence for the light chain. I'm just going to go scroll down. Okay, and then we'll go on to the CD28 hinge. So this hinge region provides a lot of flexibility for the cell, and that's not the cell, the receptor, and it allows the CAR NK cell to effectively like maneuver around the receptor to get a higher chance of being able to detect COVID-19 infected cells. So after this, we have the CD28 transmembrane domain. So this is the region that's going to go through the cell membrane. And if you know about cell membrane structure, you know that the inside of the cell membrane is hydrophobic. So we need this. We need all the amino acids here to be hydrophobic as well. So that's why it has this specific structure. And it's really helpful to take inspiration from other proteins. So this one is based on CD28, which is another type of receptor found on immune cells. So that's how we got this one. And then next we have a CD28 co-stimulatory domain. So this is part of a bigger branch of the stimulatory domains, which are going to tell the NK cell's nucleus that it needs to release these specific chemicals. And basically, this is the co-stimulatory domains are trying to enhance the signal and being able so that once the CAR NK cell detects COVID-19, it's quickly able to release those chemicals. So this is one of the co-stimulatory domains. 4-1BB is also one of the co-stimulatory domains. And after that, we have CD3 zeta. stimulatory domain. So this is the actual signaling domain that's going to send those signals, and it's based off CD3 zeta. So this is another protein. And then finally, we have our SV40 poly A signal. So this is basically going to tell the sequence. This is going to be, I'll let the sequence like translate properly and all that other stuff. So that's basically what the process is. So this is based off SV40, which is another type. It's actually a type of virus, which is able to effectively translate, and we just took inspiration from that. So that's the full car sequence. As you guys can see, it genes like it's a very complicated process, but it's actually not too hard to get these sequences yourself. I used different research papers and different articles to get sequences. I also use Benchling's gene database tool searcher, which allows you to grab genetic sequences. And another big tool is UniProt and NCBI gene database, which have so many proteins that I used, and can't thank them enough for providing these sequences to the public, so we people like me can work on stuff like this. And yeah, that's basically my whole process. My next steps are probably going to be creating primers so scientists can clone this specific DNA sequence once they get a hold of it. So I need to design primers, restriction enzyme cut sites, which I still need to learn about. So I'll let you guys know about that. And for now, that's going to be it. So thank you for listening. This is obviously really exciting and it has a lot of applications in the future. It might be a little bit overkill for like COVID-19, but it's basically just practice to get used to building this process so we can actually get up to the higher things like cancer. So thank you for listening, and I'll see you guys next time. Bye.